Welcome everyone. We are going to look at whole body coordination patterns of metabolic transcriptomes. This is opposed to, say, the new technology that allows us to look at a single tissue intercellular coordination. Uh, there has been work on intertissue coordination with a specific goal in mind. For example, examining the endocrine system. However, we are going to look at it at a systems level. Why is this important? Well, among other reasons, is that certain diseases um, occur in different multiple organs, and the organs seem to be unrelated. And uh, maybe by applying this methodology, we can infer why there are comorbidities that in certain diseases. That this is our methodology. We started with the GTEx dataset, which includes over 50 tissue types and nearly 1,000 samples, and includes multiple samples per donor, so we can look at the donor-specific correlations within different tissues. We applied the WGCNA algorithm to cluster the genes on a tissue per tissue basis, and we also applied KEGG pathways to infer what the module function is. Then we applied statistical analysis to see how much um, the metabolic modules differ from randomly selected modules to see if we can observe statistical significance. So let's look at the cohort 2 specifically. The y-axis shows us the number of pairwise correlations per threshold. The x-axis shows us the thresholds. And the dark green line shows us the number of positive correlations that were metabolic. The shaded green regions were from 1,000 randomly selected samples of similar composition and size to the metabolic modules, how often the 95th percentile and 90th percentile in regions were in terms of positive metabolic correlations, and we see that at the lower correlation thresholds up until 0.2 something, the metabolic modules exceed a randomly selected set. The negative metabolic modules in general appear in both cohort 2 and cohort 1 to be below the average. Uh, Now the results are more dramatic when we take the positive over the negative ratio, and then we see that among the metabolic, we clearly exceed the 95th percentile of the average random sampling in nearly all correlation thresholds on both cohorts. Uh, when we look, rather than at the correlation threshold, we look at the p-value threshold, then we can also include a line that shows us the false discovery rate, which in both graphs is well below any of the measures, meaning that even the among the random set of correlations, the correlation the modules, the correlations themselves are not at all random. Now we also did analysis on a click-based analysis, and we used three metrics. So one is the max degree, and in this instance, it's liver 38 in the metabolic module, and we see that it is above the 95th percentile uh, when you compare the largest click size in a random set. When you measure the largest click size, we got 12, and potentially the largest it could be is 40. Uh, in practice, among the randoms, we got a largest of 2, and with this metric, we hit the 95th percentile. And when we did the mean community size of the largest click, we got a value of 6, which also exceeds the 95th percentile. To summarize, the metabolic positive modules exceed intertissue correlations exceed the a randomly selected pairwise set. The positive to negative ratios uh, of metabolic are larger than the random set. The random set tends to be approximately 1 to 1, and we also see that the clicks of the metabolic set are typically larger. Thank you very much.